Today we will discuss 2-phosphoglycolate, a phosphorespiratory metabolite. 2-phosphoglycolate is a 2-carbon compound that is produced in large quantities by oxidative damage to the carbon dioxide acceptor molecule, ribulose 1,5 bisphosphate. Rubisco enzyme catalyzes the addition of carbon dioxide or oxygen to ribulose 1,5 bisphosphate. 2-PG or 2-phosphoglycolate is also generated during DNA end repair and is a product of pyruvate kinase side reactions. It is toxic to plants and bacteria and inhibits several metabolic pathways, including triose phosphate isomerase, phosphofructokinase, and pseudohaptulose 1,7 bisphosphate phosphatase. Phosphoglycolate phosphatase enzyme dephosphorylates the 2-PG and converts it to glycolate, which can then be incorporated into general metabolism. Today we will discuss how 2-PG is formed, its effects on plants, and its relationship with photorespiration. 2-phosphoglycolate is an important metabolite in many organisms. In photosynthesis, carboxylation, and oxygenation. Both reactions are catalyzed by the enzyme ribulose 1,5 bisphosphate carboxylase oxygenase, that is Rubisco. In carboxylation, which is the first step in the photosynthetic carbon reduction cycle, it involves the addition of carbon dioxide. Just look at this diagram. Enzyme Rubisco, carboxylation, and oxygenation. Carboxylation is the first step in the photosynthetic carbon reduction cycle, right? It involves the addition of carbon dioxide and water to ribulose 1,5 bisphosphate. And this process produces 2 molecules of 3-phosphodeglycerate, that is 3-PGA, right? In oxygenation, it involves the addition of oxygen and water to ribulose 1,5 bisphosphate and producing 1 molecule of 3-PG and 1 molecule of 2-phosphoglycolate, right? 3-PGA enters the Calvin cycle, normal Calvin cycle, but the 2-carbon compound, that is 2-PG, is a dead-end metabolite. In plants, 2-PG is converted to glycolate, and glycolate can then be incorporated into general metabolism. If accumulated, 2-PG is a potentially toxic metabolite that can have harmful effects on plants and bacteria. In plants, it can inhibit several metabolic pathways. 2-PG can also sequester large amounts of carbon and phosphorus, draining the Calvin-Benson cycle's metabolite pool, then strongly inhibit photosynthesis, prevent carbon allocation towards starch biosynthesis, impede the biosynthesis of phosphorylated intermediates, and triose phosphate export from the chloroplast. On the other hand, 2-PG is also an important metabolite in many organisms. In mammals, it activates the 2,3-bisphosphoglycerate phosphatase enzyme module of the erythrocyte-specific trifunctional enzyme bisphosphoglycerate mutase. Binding of 2-PG stimulates the phosphatase activity of this enzyme, that is bisphosphoglycerate mutase, up to 1600-fold, lowering the content of 2,3-bisphosphoglycerate, which dissociates from and increases the affinity to oxygen of hemoglobin. 2-PG is produced by oxidative damage to the carbon dioxide acceptor molecule, that is ribulose 1,5-bisphosphate in plants. The resulting ribulose 1,5-bisphosphate damage is repaired in the multi-step photorespiratory pathway via the conversion of 2-PG to 3-phosphoglycerate, 3-PG. And we know 3-PGA is the actual product of ribulose bisphosphate carboxylation, from which ribulose 1,5-bisphosphate is eventually regenerated in the Kelvin-Benson cycle. The photorespiratory pathway is actually a metabolic repair system that enables the carbon dioxide fixation enzyme Rubisco to sustainably operate in the presence of oxygen. In energetic terms, photorespiration is inarguably wasteful in that it is costly in terms of carbon dioxide and ATP losses, right? But photorespiration is necessary because it removes an auto-inhibitory metabolite, 2-phosphoglycolate, that is 2-PG, to avoid clasp of metabolism and recycled as efficiently as possible. The breakdown of 2-PG is catalyzed by the enzyme 2-PG phosphatase, which are metabolite damage repair enzyme with multiple functions, right? 2-PG phosphatase is a damage repair enzyme with multiple functions. In oxygenic autotrophs, they catalyze the first step of the photorespiratory pathway, and in mammals, yeast, and possibly other organisms, they remove 2-PG and destroy toxic side products of glycolysis. The conversion process releases one molecule of photorespiratory carbon dioxide per every 2 recycled 2-PG molecule, right? Thereby reducing net photosynthesis. This is why over the last decades, photorespiration has been seen as an attractive target for the breeding of high-yield crops. Thanks for watching. If you liked this video, make sure to subscribe for more. Thank you.